Welcome to the Deep Dive, where we take a stack of sources and extract the absolute most important nuggets of knowledge for you to truly master a topic. Today we're plunging into one of the most intricately controlled aspects of human physiology, the regulation of body fluid osmolality and volume.
Yeah, it's fascinating stuff.
Imagine your body not just as an aquarium, but maybe more like a meticulously engineered bioreactor, where every single cell needs the perfect, precise balance of water and salts to function optimally.
Absolutely. Any deviation, too much, too little, too concentrated, or too dilute, and the whole system can be thrown into disarray.
So today we're going to uncover how your kidneys, your brain, and this whole symphony of hormones coordinate to maintain this exquisite balance, giving you an understanding that really goes beyond the surface level.
That's right. Our mission today is to truly understand the incredible precision, really, with which your body manages its internal water and salt levels. We'll be examining the fundamental mechanisms, delving into the cellular events, the systemic regulation, basically everything that ensures every sip of water and every salty bite is accounted for.
And the kidneys are central.
Oh, completely. The kidneys, as we'll see, are the ultimate arbiters. They're constantly fine-tuning excretion to maintain a stable internal environment homeostasis. By the end of this deep dive, you'll possess, well, a professional-level grasp of this vital physiological system.
We've compiled a formidable stack of detailed physiological sources to guide us through these renal mechanisms, water, and NaCl excretion. Get ready to connect some serious dots and truly understand the elegant complexity that keeps you going. Okay, so to begin our journey, let's establish the foundational concepts. What exactly is body fluid osmolality, and how does water actually get into and out of our system?
Right, the basics first.
When we consider water gain, the primary source obviously is oral intake, not just liquids, but also the water content within solid foods.
People sometimes forget about the water in food.
Exactly. And beyond that, a significant amount of water, maybe around 10%, is actually produced metabolically when our bodies break down ingested foods, particularly carbohydrates and fats.
Right, metabolic water.
And then in clinical scenarios, especially during illness, intravenous fluids become a really crucial regulated route for water supply.
Definitely. Conversely, water loss happens via several distinct routes. The kidneys are the major route for water excretion under most conditions. And critically, this renal excretion is exquisitely regulated to maintain overall water balance.
So they're the main regulators.
They are. The gastrointestinal tract also contributes, though typically only a small amount, maybe 100 milliliters per day in feces. But, and this is important, this loss can dramatically increase under pathological conditions like severe diarrhea.
Like cholera, the numbers are staggering there.
Exactly, staggering, like 20 liters per day potentially. Or, of course, with persistent vomiting.
And then we have sweat. That's an active process involving controlled elimination of water and electrolytes through specialized glands.
Right.
These three kidneys, GI tract, and sweat represent what we categorize as sensible water loss because, well, you're basically aware of it happening.
You feel it or see it.
But there's also insensible water loss. This occurs continuously, passively, through evaporation from your skin and your respiratory passages during breathing. You're completely unaware of it.
It just happens.
What's truly critical to appreciate is how things like the environment and physical activity profoundly impact these loss routes. For example, hot weather or, say, sustained heavy exercise.
Yeah, big changes then.
Right. Both sweat production and water loss from the lungs via respiration can increase dramatically. And in response, the kidneys adapt by significantly decreasing urine volume to conserve water. Although it's important to note that renal conservation alone often can't fully compensate for extreme conditions.
No, intake becomes critical then. This brings us to osmolality, which is essentially a measure of the solute concentration in a solution.
How many bits are dissolved in the water, basically?
Pretty much. And body fluid osmolality is, in fact, one of the most rigorously regulated parameters in human physiology. It's kept within an extremely narrow range, typically 280 to 295 millosm per kilogram of H2O.
That's a tight range.
It is incredibly tight. The kidney's central and indispensable role here is to simultaneously regulate both osmolality and total fluid volume by meticulously controlling the excretion of water and sodium chloride.
Yeah.
Our sources show that a healthy individual has this remarkable ability to vary their urine osmolality, or UOSM, over a huge range from as low as maybe 50 millosms per kilogram of H2O.
Which means very dilute urine, lots of it.
Exactly. Large volume dilute up to maybe 18 liters a day. Or they can go as high as about 1,200 millism per kilogram of H2O.
Super concentrated, minimal volume.
Right, producing a small volume of highly concentrated urine, maybe as little as half a liter per day. This crucial, precise ability of the kidneys to regulate water excretion independently from total solute excretion, separately from things like sodium, potassium, urea, it's not just important, it's absolutely fundamental for survival.
Okay, so this is where it gets really fascinating and maybe a bit counterintuitive, especially if you're aiming for that professional mastery. If your serum sodium concentration is off, right outside the normal range, your immediate thought might be, okay, it's a salt problem.
That's the common assumption.
But here's the crucial insight. More often than not, an abnormal serum sodium level is actually a primary disorder of water balance, not necessarily a direct problem with sodium itself.
Exactly. This is a key diagnostic point. Changes in total sodium balance primarily alter the extracellular fluid volume, the ECFV, not its osmolality.
Okay, so salt changes volume, water changes concentration.
Broadly speaking, yes. That's a critical distinction because the body prioritizes maintaining osmolality, that concentration, even if it means altering the overall fluid volume.
Got it.
Consider this example from our sources. If you have a positive water balance, say you add 1 liter of pure water to your system, this initially increases your ECFV, reduces its osmolality, and consequently decreases your plasma sodium concentration hyponatremia.
Right, diluted sodium.
Precisely. The kidneys, driven by the need to restore osmolality, respond by excreting that exact liter as hypososmotic or dilute urine, bringing the system back into balance.
Okay.
Conversely, with negative water balance, if you lose 1 liter of water, your ECFV decreases, its osmolality increases, and your plasma sodium concentration rises, hypernatremia.
Concentrated sodium.
Right. The renal response is then to conserve water by excreting a minimal volume of hyperosmotic concentrated urine. And at the same time, thirst is powerfully stimulated to compel you to drink and restore fluid balance.
And when these plasma osmolality changes happen quickly, particularly acutely, the effects can be really dangerous, right? Primarily hitting the brain.
Absolutely. If plasma osmolality falls rapidly, water shifts osmotically into brain cells, causing them to swell. This cellular swelling can lead to a cascade of neurological symptoms, things like nausea, malaise, headaches, then confusion, lethargy, seizures, even coma in severe cases.
And the reverse, if osmolality goes up quickly.
Same problem, opposite direction. Water gets drawn out of the cells, causing them to shrink. This also predominantly affects the brain, leading to similar severe symptoms: lethargy, weakness, seizures, coma, potentially even death.
But there's a key clinical detail here, isn't there? About timing.
Yes, very important. If these changes occur more gradually, say over days or weeks, brain cells actually have remarkable compensatory mechanisms. They can actively adjust their internal osmolas, either eliminating them or generating new ones to minimize these volume changes.
So the symptoms might be less severe, or maybe not even apparent.
Exactly, which can make diagnosis trickier in those chronic cases.
Okay, so if the kidneys are the main effectors, doing the work of adjusting water excretion, what's the master regulator pulling their strings? This brings us to arginine vasopressin, or AVP.
Also known as ADH, antidiuretic hormone.
Right, the famous ADH. This small but mighty nonapeptide is synthesized with incredible precision in specific neurosecretory nuclei within the hypothalamus, the supraoptic nucleus, SON, and the paraventricular nucleus, PDN. AVP exerts its powerful effects primarily by acting through V2 receptors, which are strategically located in key nephron segments within the kidney.
And the effect is quite dramatic.
It really is. If your circulating AVP levels are low, you excrete a substantial volume of dilute urine, a diuretic effect. Conversely, when AVP levels are high, your kidneys produce a small volume of highly concentrated urine, a powerful antidiuretic effect.
To really master this, let's trace AVP's journey right down to the cellular level. The gene for AVP is on chromosome 20, and it codes for a larger precursor molecule, a pre-prohormone. This undergoes a crucial initial cleavage step in the rough ER, where a signal peptide is removed. Then, once it's packaged into neurosecretory granules, further enzymatic cleavage happens, transforming it into the active AVP molecule, along with neurophysin, that's a carrier protein, and a glycopeptide called copeptin.
So it's processed along the way.
Exactly. These granules are then actively transported down the axons of these hypothalamic neurons all the way to their nerve endings in the posterior pituitary gland. They're stored there, basically ready for release.
And how does release happen?
When these neurons get the signal to secrete AVP, an action potential reaches the nerve terminal. This triggers the opening of voltage-gated calcium channels. Calcium rushes into the cell, raising intracellular calcium concentration, which then activates the exocytotic machinery. The granules fuse with the cell membrane and release AVP into the bloodstream.
It's a very controlled process. Now, the control of AVP secretion itself is remarkably sophisticated. It's primarily governed by two major physiological stimuli, osmotic and hemodynamic.
Right, concentration and pressure volume.
But it's important to recognize other factors play a role too. Things like nausea, acute hypoglycemia, even angiotensin-aldosterone can powerfully stimulate AVP release.
And conversely, ANP, atrial natriuretic peptide, ethyl alcohol, and antiemetic drugs tend to inhibit it. Oh, and nicotine is a known stimulator too.
Interesting. But osmotic control is really the dominant player, right? The most sensitive.
Without question. Specialized neurons called osmoreceptors, located in the OVLT, that's the organum vasculosum of the lamina terminalis in the hypothalamus, are exquisitely sensitive to tiny changes in body fluid osmolality, especially changes caused by effective solutes like sodium and its anions.
What makes a solute effective versus ineffective?
Good question. Effective solutes like sodium don't easily cross cell membranes, so they create an osmotic gradient that pulls water. Ineffective solutes like urea or glucose generally cross membranes more freely, so they don't cause significant water shifts or cell volume changes that the osmoreceptors would detect strongly.
Got it. So sodium changes are key.
Exactly. When effective plasma osmolality increases, these osmoreceptor cells sense the higher concentration outside and they shrink. This shrinkage activates specific, non-selective cationic channels in their membrane, letting positive ions flow in, which depolarizes the cell.
Okay.
This depolarization triggers action potentials that travel to the AVP-synthesizing neurons in the SO and PVN, telling them to release AVP.
And the opposite happens if osmolality drops.
Precisely. A decrease in effective plasma osmolality profoundly inhibits both AVP synthesis and secretion, and AVP itself is rapidly degraded in the plasma. Its half-life is only a few minutes. This allows its levels to drop very quickly, which is critical for that precise on-demand regulation.
And there's some newer thinking about how these osmoreceptors actually work, right? Something about stretch.
Yes, that's fascinating recent data. It suggests it might actually be cell membrane stretch rather than overall cell volume change that directly determines osmoreceptor activity. This could be mediated by specific TRPV channels, TRPV1, 2, and 4, which are sensitive to membrane stretch and might help inactivate the receptors in low osmolality states. Still an active area of research, but very interesting.
This coordinated interplay establishes a remarkably precise threshold or set point for AVP release. On average, it's around 280 mosmol per kilogram of H2O. What's truly remarkable and really speaks to the system's sensitivity is that a mere 2% to 4% increase in plasma osmolality above this set point can trigger the maximum antidiuretic effect. Below this threshold, virtually no AVP is released.
It's incredibly fine-tuned.
And while this set point is generally stable, it can shift, right, like during pregnancy.
Yes, it can dynamically decrease in certain physiological or pathological states. Pregnancy is a key example, also the menstrual cycle in response to estrogen, or following a significant drop in blood pressure or major blood loss. The shift in pregnancy, for instance, is thought to be linked to hormones like relaxin and HCG.
Beyond osmotic control, hemodynamic changes, so alterations in blood volume or pressure, also exert powerful control over AVP, though typically less sensitive.
That's right. This control is mediated by baroreceptors located in two key areas. The low-pressure cardiopulmonary circuit, think cardiac atria, large pulmonary vessels, which respond to the overall fullness of the vascular system, and the high-pressure arterial circuit, found in places like the aortic arch and carotid sinus, responding to arterial blood pressure. Both types are sensitive to the mechanical stretch of the vessel walls and send signals via the vagus and glossopharyngeal nerves to the brainstem, which then relays them to the AVP cells in the hypothalamus.
And it takes a bigger change to trigger this pathway.
Generally, yes. Typically, a 5% to 10% decrease in blood volume or pressure is needed to stimulate AVP via these baroreceptors compared to that tiny 2–4% change in osmolality. Our sources also mention that things like bradykinin and histamine, which cause vasodilation and lower blood pressure, can stimulate AVP, while norepinephrine, which raises blood pressure, tends to inhibit it.
And here's where the body's design really optimizes for survival, integrating these two systems. When there's a decrease in blood volume or pressure, a serious threat.
Right, a dangerous situation.
The AVP set point actually shifts to a lower osmolality, and the slope of the relationship gets steeper. What does that mean functionally?
It means that in critical situations, like circulatory collapse, your kidneys will prioritize conserving water and maintaining vital blood volume, even if it means letting the body fluids become slightly less concentrated, slightly hyposmonotic. Survival trumps perfect osmolality in that scenario.
Makes sense. And the opposite if volume or pressure increases.
Yes, then the set point shifts higher and the slope decreases, allowing the body to get rid of excess water more easily. It's an incredibly adaptive and finely tuned system for resilience.
Okay, this inevitably leads to the question, what happens when this delicate AVP system goes wrong?
Ah, yes, the clinical implications. One important condition is central diabetes insipidus, or CDI. This results from insufficient AVP release from the posterior pituitary.
So the signal isn't getting sent properly.
Exactly. While there are rare inherited forms due to AVP gene mutations, it's more commonly acquired maybe from head trauma, brain tumors, infections affecting the hypothalamus or pituitary.
And the symptoms.
Profound polyuria, meaning huge volumes of dilute urine, and intense polydipsia, that constant thirst, trying to compensate for the massive water loss. If they can't get water, they rapidly become hyperosmotic, severely dehydrated.
But it's treatable.
Yes, thankfully. The concentrating defect can be effectively corrected by giving exogenous AVP, or, more commonly, a synthetic analog like desmopressin, DDAVP.
Then there's the flip side, syndrome of inappropriate ADP secretion, or SIADH.
Right. SIADH is relatively common and clinically very significant. It's characterized by plasma AVP levels that are inappropriately high, they're elevated even when osmolality is low, and volume pressure are normal.
What causes that?
A wide range of things, many medications, NSAIDs, antidepressants, anti-seizure drugs, antipsychotics, some anti-tumor drugs. Also, certain cancers, especially small cell lung carcinoma, can ectopically produce AVP themselves.
And the result is water retention.
Yes. Water retention, leading to progressively hyposmotic body fluids, hyponatremia.
Yeah.
And here's a key diagnostic clue: urine that's paradoxically more concentrated than you'd expect given their low body fluid osmolality.
How is SIADH treated?
Often involves AVP receptor antagonists.
Yeah.
These drugs block the V1A and V2 receptors, inducing what's called a choresis, basically. Targeted water excretion without losing lots of electrolytes helps get rid of the excess water.
Okay, distinct from SIADH and CDI is nephrogenic diabetes insipidus, NDI. What's the issue there?
In NDI, the collecting ducts of the kidney simply don't respond normally to the circulating AVP. The signal is there, but the kidney isn't listening.
So same symptoms as CDI, polyuria, polydipsia?
Exactly the same symptoms, but a different underlying cause. NDI can be acquired, often due to decreased expression of the AQP2 water channels in the collecting duct. Common causes include low potassium, hypokalemia, lithium treatment.
Right, you mentioned a high percentage of lithium patients develop this.
Yeah, around 35% develop some degree of NDI. Also, urinary tract obstruction, a chronically low protein diet, high calcium, hypercalcemia.
And inherited forms.
Also exist. About 90% of hereditary NDI comes from mutations in the AVPR2 gene, the gene for the V2 receptor, which is X-linked. The other 10% comes from mutations in the AQP2 gene itself, which can be autosomal recessive or, very rarely, dominant.
Is there a difference in how those AQP2 mutations cause NDI?
Yes, interestingly. The recessive mutations often affect the pore-forming region of AQP2, preventing proper channel assembly. The dominant mutations usually affect post-translational modifications, like phosphorylation, which messes up the trafficking or function of the channel, even if it's formed correctly.
Wow, specific. And then there's one more twist. Nephrogenic syndrome of inappropriate antidiuresis, NSIAD.
Right, NSIAD. This is a genetic disorder caused by activating or gain-of-function mutations in that same AVPR2 gene. These mutations make the V2 receptors constitutively active. They're basically stuck in the on position, even without AVP.
So the kidney acts like AVP is high, even when it's not.
Precisely. Individuals with NSIAD present with findings very similar to SIADH: reduced plasma osmolality, hyponatremia, inappropriately concentrated urine. But here's the absolute crucial distinction for diagnosis. Unlike SIADH, where AVP levels are high, people with NSIAD have undetectable levels of AVP in their plasma. The kidney's water channels are constantly open, but the hormonal signal isn't there.
That's a critical difference. Okay, so now that we understand AVP's importance and what happens when it goes wrong, let's zoom in even further. What exactly is AVP doing at a cellular level in the kidneys to cause these dramatic shifts in urine production?
AVP has three primary interconnected actions on the kidneys that are crucial for water balance. First, and most importantly, it profoundly enhances water reabsorption from the tubular fluid, specifically in the late distal tubule and the entire collecting duct system, by dramatically increasing their water permeability.
Okay, makes them leaky to water.
Exactly. Second, it significantly increases the permeability of the inner medullary part of the collecting duct to urea.
We'll come back to why urea is important later, I assume.
Absolutely, it's key for concentration. And third, AVP stimulates sodium chloride reabsorption in the thick ascending limb of Henle's loop, the distal tubule, and the collecting duct.
Okay, let's focus on the water action first. How does it increase water permeability at the cellular level?
All right, let's dissect that cellular mechanism. The V2 receptor, encoded on the X chromosome, is a classic G protein-coupled receptor on the basolateral membrane, the blood side of the principal cells in the distal tubule and collecting duct.
Okay.
In the basal state, without AVP, the apical membrane, the side facing the urine, is largely impermeable to water. Why? Because very few AQP2 water channels are actually present on that membrane. They're stored inside the cell in vesicles.
Waiting for the signal.
Exactly. When AVP binds to its V2 receptor, it kicks off a signaling cascade. It activates adenyl cyclase, which rapidly increases intracellular cyclic AMP, or CANT-MP. This CAMP surge then activates protein kinase A, or PKA.
The classic CAMP pathway.
Precisely. PKA then does two key things. It phosphorylates those AQP2 water channels, essentially tagging them for action, and it also increases the transcription of the AQP2 gene itself, contributing to longer-term adaptation.
So this is where the rapid on-demand water reabsorption happens.
Yes. The vesicles containing those phosphorylated, primed AQP2 channels then move along the cell's microtubule network using a motor protein called dynin towards that apical membrane.
Okay.
Once they get there, snare proteins help them fuse with the plasma membrane, effectively inserting the AQP2 channels into it. This sudden insertion dramatically increases the apical membrane's water permeability.
And water flows in.
Right. Water rapidly enters the cell from the tubular lumen, driven by a strong osmotic gradient. The urine in the tubule is less concentrated than the cell interior. Once inside, water exits across the basolateral membrane into the interstitial fluid via other aquaporin channels, AQP3 and AQP4, which are always present there.
So AQP2 is the regulated one on the apical side, AQP3 and 4 are constitutive on the basolateral side.
You got it. And when AVP is no longer bound, either because levels drop or it's degraded, the AQP2 channels are swiftly removed from the apical membrane by clathrin-mediated endocytosis. This makes the membrane impermeable to water again. These removed AQP2s are either recycled and stored for reuse or targeted for degradation.
It's a very dynamic system. Does AVP also have longer-term effects?
Yes, absolutely. Beyond this rapid trafficking, AVP also regulates the chronic expression, the overall abundance of both AQP2 and AQP3. For instance, if someone chronically drinks huge amounts of water, like in psychogenic polydipsia,
Okay.
the sustained low AVP leads to reduced abundance of both AQP2 and AQP3. So if they suddenly restrict water, they can't maximally concentrate their urine because they simply lack enough water channels.
And the opposite, chronic dehydration.
Right. Chronic water restriction leads to sustained high AVP, which increases the expression of AQP2 and AQP3 protein. This significantly enhances their capacity to make maximally concentrated urine. We also see changes in disease states. AQP2 expression is often reduced in conditions that impair concentrating ability, like hypercalcemia or hypokalemia. And it's often increased in states of water retention, like heart failure, cirrhosis, or pregnancy, reflecting the body's attempt to hold on to water.
Okay, you mentioned AVP also affects urea reabsorption. Why is that important?
It's absolutely critical for urine concentration. AVP increases the permeability of the terminal part of the inner medullary collecting duct to urea. This boosts urea reabsorption into the medullary interstitium, the deep tissue of the kidney.
And that helps make the medulla really concentrated.
Exactly. It's essential for establishing and maintaining the high osmolality of that medullary interstitial fluid. Without high urea concentration there, maximal urine concentration just isn't possible.
How does AVP increase urea permeability?
The collecting duct cells have specific urea transporters, UTA1 on the apical membrane and UTA3 on the basolateral side. AVP, acting via that same CAMP-PKA pathway, directly increases the expression of both UTA1 and UTA3, enhancing urea transport out of the tubule. Interestingly, high osmolality in the medulla itself can also increase urea permeability, likely through a different pathway involving phospholipase C and protein kinase C, another layer of regulation.
And finally, you said AVP stimulates sodium chloride reabsorption.
Yes, it does. It activates key NaCl transporters in various segments. The NKCC2 supporter in the thick ascending limb, the Na plus Cl supporter in the distal tubule, and the epithelial sodium channel, ENaC, in the late distal tubule and collecting duct.
Why stimulate salt reabsorption if its main job is water?
It's thought that this stimulation, particularly in the thick ascending limb, plays a crucial role in maintaining and even augmenting that hyperosmotic medullary interstitium we just talked about. More salt pumped out there helps create the gradient needed to pull water out of the collecting duct later on. It's all interconnected.
Okay, this is really where the kidney showcases its, I don't know, unparalleled engineering genius. Its ability to separate water from solute to make urine that can be incredibly dilute or incredibly concentrated.
It's truly remarkable.
The core principle is that the kidneys must be able to excrete urine that's either hypoosmotic, dilute, or hyperosmotic concentrated. And this absolutely requires separating solute from water at specific points along the nephron.
Right. The proximal tubule doesn't really do the separation. It reabsorbs water and solutes proportionally, so the fluid stays isosmotic.
So where does the magic happen?
The loop of Henle, particularly its thick ascending limb, is the major functional site for separating solute and water. It's indispensable for producing both dilute and concentrated urine.
Okay, let's walk through making dilute urine first. Water diuresis. What's the goal?
The fundamental goal is to efficiently reabsorb solute while preventing water reabsorption. And critically, this assumes low or absent AVP levels.
Right, AVP is off.
Okay, so isosmotic fluid enters the loop from the proximal tubule. As it goes down the descending thin limb, water is reabsorbed via AQP1 channels, mainly in the outer medulla. This makes the tubular fluid inside more concentrated, hyperosmotic.
Okay.
Then, as the fluid enters the inner medulla and loops back up into the thin ascending limb, this segment is notably impermeable to water, no AQP1. But it does reabsorb NaCl either passively or actively, starting the crucial process of diluting the tubular fluid.
This dilution process gets more powerful in the thick ascending limb, right?
Absolutely. The thick ascending limb is also largely impermeable to water, but it vigorously and actively pumps NaCl out of the tubular fluid. This further dilutes the fluid inside. That's why we call it the diluting segment. The fluid leaving the thick ascending limb is distinctly hyposmotic compared to plasma.
And if AVP is still absent?
Then the distal tubule and cortical collecting duct, which lack AQP2 on their apical membranes without AVP, also remain impermeable to water, but continue to actively reabsorb NaCl. This dilutes the fluid even more.
What about the final segment, the medullary collecting duct?
Even without AVP, it has a slight baseline permeability to water, so a little bit might be reabsorbed there, but not much. The impressive end result: urine that can be as dilute as maybe 50 millism per kilogram of H2O, very low in NaCl, and produced in huge volumes up to 18 liters per day.
Wow. Okay, now let's flip it. Producing hyperosmotic concentrated urine antidiuresis. This happens when AVP is high.
Correct. Here, the kidney's objective is to remove water from the tubular fluid without removing proportionate amounts of solute. And this absolutely requires that powerfully hyperosmotic medullary interstitium we talked about. That's the engine.
It acts as the osmotic sink, pulling water out.
Precisely. The initial steps in the loop of Henle are qualitatively similar. NaCl is reabsorbed from the ascending limbs, initially diluting the fluid. However, and this is key, that reabsorbed NaCl now accumulates in the medullary interstitium, significantly raising its osmolality. And remember, AVP itself stimulates this NaCl transport. This buildup of salt provides the essential driving force for water reabsorption later.
Okay, so the loop sets up the gradient. What happens next?
The fluid, still somewhat hyposmotic, enters the distal tubule and cortical collecting duct. Because AVP levels are high, water permeability here dramatically increases due to that mass insertion of AQP2 channels onto the apical membrane.
Right, the gate's open.
Exactly. Water rapidly diffuses out of the tubule into the interstitium, following the osmotic gradient. The tubular fluid osmolality increases, reaching about 290 millism kilogre H2O, basically isosmotic with plasma by the time it exits the cortical collecting duct.
So it equilibrates with the cortex, then it goes deeper into the medulla.
Yes. As this now isosmotic fluid descends deeper into the medulla within the medullary collecting duct, water continues to be reabsorbed, pulled out by that progressively hyperosmotic interstitium created by NaCl and urea. This process can concentrate the fluid way up to a maximum of maybe 1,200 ml/7 kg H2O at the very tip of the papilla.
Incredible concentration.
The profound result is urine with an osmolality up to 1,200 ml/7 kg H2O, rich in urea and other non-reabsorbed solutes, and produced in a minimal volume, as low as half a liter a day.
So it seems like a relatively constant volume of dilute fluid gets delivered to the AVP-sensitive part.
That's a key point.
And then plasma AVP levels dictate how much water gets pulled out.
Exactly. And during antidiuresis, most of that water reabsorption actually occurs before the fluid reaches the deepest part of the medulla in the distal tubule, cortical, and outer medullary collecting ducts. This means a smaller volume of already concentrated fluid reaches the inner medullary collecting duct. That's a crucial design feature. It minimizes water entry into the deepest medulla, helping to preserve that vital hyperosmotic gradient needed for maximal concentration.
That hyperosmotic medullary interstitium really is the hero here. What makes it up again?
Its principal solutes are strategically accumulated sodium chloride and urea. They create this profound osmotic gradient, increasing progressively from the cortex, isotonic, down to the papillary tip, hypertonic. Other solutes like ammonium and potassium add a bit, but NaCl and urea are the stars.
And at the tip, it's about 1,200 millism in total.
Roughly, yes. And interestingly, about half of that, maybe 600 millism, is from NaCl, and the other half, another 600 millism, is from urea.
How does the NaCl gradient get set up?
That's fundamentally generated by the countercurrent multiplication mechanism of Henle's loop specifically, the active pumping of NaCl without water by the thick ascending limb.
And the urea accumulation, you said, was more complex.
It is. It's most effective during antidiuresis and involves urea recycling. Remember, AVP increases UTA1 and UTA3 transporters in the medullary collecting duct.
Right, pulling urea out into the interstitium.
Exactly. Some of that reabsorbed urea is then strategically secreted back into the thin descending limbs via UTA2 transporters, and some also enters the vasorecta capillaries via UTB. This continuous trapping, cycling, and recirculation allows urea to reach those incredibly high concentrations, up to 600 millimole at the papilla tip.
And that's critical because urine can't get more concentrated than the papillary interstitium.
Absolutely fundamental rule. Any reduction in that medullary interstitial osmolality directly impairs the kidney's ability to maximally concentrate urine.
What about the vasorecta, the capillaries down there? What's their role?
They are equally fascinating and crucial. These are specialized capillary networks supplying the medulla. They're uniquely highly permeable to both solute and water, and they form their own hairpin loops parallel to Henle's loop.
They have a dual role.
Yes, vital. First, they deliver oxygen and nutrients to the metabolically active cells down there. But more importantly, their countercurrent exchange mechanism allows them to remove the excess water and solute that are constantly being added to the interstitium, without washing out that crucial osmotic gradient.
How does flow affect them?
It's exquisitely flow dependent. If medullary blood flow increases too much, it can wash out the gradient, impairing concentration. Conversely, if flow is severely reduced, you don't get enough oxygen delivery, which impairs the active salt transport needed to maintain the gradient. So it's a balance. The kidneys meticulously maintain this gradient, steeper during antidiuresis, flatter during diuresis.
Okay, so how do clinicians actually assess renal diluting and concentrating ability?
It requires precise measurements of urine osmolality and volume. As we said, the normal range is huge, 50 up to 1,200 millimini H2O, corresponding to urine output from 18 liters down to half a liter.
And it hinges on separating water from solute.
Exactly. The kidney's ability to do this generates what we call solute-free water. This is either deliberately excreted in dilute urine or meticulously conserved and returned to the body in concentrated urine.
To achieve maximal dilution, that 18 liters a day, what conditions are needed?
Several strict conditions must be met. First, AVP must be completely absent. No AVP, no significant water reabsorption in the collecting duct.
Okay.
Second, the tubular structures responsible for dilution, the ascending thin limb, thick ascending limb, distal tubule, cortical collecting duct, must all be functioning normally to achieve that solute water separation.
Makes sense.
And third, there must be adequate continuous flow of tubular fluid delivered to these diluting segments. If GFR is too low or too much fluid is reabsorbed proximally, you just don't deliver enough raw material to the diluting segments to generate maximal free water.
Okay. Now, conversely, for maximal concentration, getting down to that 0.5 liters a day.
Similar critical conditions apply, highlighting the integrated function. First, Adequate delivery of tubular fluid to the separating segments, especially the thick ascending limb, is still needed.
Right, need to flow.
Second, NaCl reabsorption by these segments, again, especially the thick ascending limb, must be robust and normal. Third, that strongly hyperosmotic medullary interstitium must be present and well maintained. Its osmolality depends on that NaCl reabsorption, effective urea accumulation, which needs adequate dietary protein, and proper vasorecta function.
And finally,
Maximum circulating AVP levels must be present, and the collecting duct system must respond normally to AVP. It's this intricate interplay that allows for profound water conservation.
the kidneys are doing incredible work, but they can't manage water balance entirely alone.
Yeah.
What's the body's primary way of knowing it needs to actually drink?
Thirst.
The fundamental drive. What triggers it?
Thirst is profoundly influenced by changes in plasma osmolality, blood volume, or blood pressure. But increased plasma osmolality is by far the most potent physiological stimulus. A remarkably small 2% to 3% increase in osmolality can trigger a strong desire to drink.
More sensitive than the volume pressure trigger.
Generally, yes. A more significant reduction in blood volume or pressure, maybe 10% to 15%, is typically needed to elicit a comparable thirst response.
Where is the thirst center?
It's located in the same region of the hypothalamus that regulates AVP secretion, and it primarily responds to those effective solutes, like sodium chloride. Interestingly, the thirst threshold, usually around 295 mSmKre H2O, is slightly higher than the AVP secretion threshold of 280.
Why is that significant?
It means thirst usually kicks in when AVP secretion is already ramping up towards its maximum. It ensures water conservation mechanisms are engaged before the body actively signals for water intake. It's an efficient design.
Does angiotensin II play a role in thirst too?
Yes, significantly. Angiotensin II directly acts on cells of the thirst center to evoke the sensation. This is particularly important when blood volume and pressure are low, helping restore fluid balance by promoting intake alongside its other actions.
Here's a curious detail. Drinking satisfies thirst almost immediately, right? Even before the water is absorbed.
That's right. It involves complex oropharyngeal and upper gastrointestinal receptors. That immediate sensation of relief, especially with cold water, is mediated locally.
But it's temporary.
Exactly. It's only transient. Full, sustained satisfaction of thirst only occurs when plasma osmolality, blood volume, or pressure are actually corrected back to their homeostatic set points.
The synergy between AVP and thirst seems crucial.
It's a perfect example of robust biological redundancy. High osmolality triggers both drinking and AVP-mediated water conservation. Low osmolality suppresses thirst, and the absence of AVP enhances renal water excretion. Works beautifully together.
But what if fluid intake isn't driven by physiology, like just habitual drinking?
Then maintaining normal body fluid osmolality becomes entirely reliant on the kidney's ability to excrete that excess water load.
Thinking about real-world examples, like those Boston Marathon runners getting hyponatremia from overhydration.
Yeah.
That really drives home how you can overwhelm the kidneys.
It's a classic, powerful example. And to really grasp the nuance there, remember that water is also produced metabolically during intense exercise from burning fuels like glycogen and triglycerides.
Ah, right. Internal water production, too.
So the total water gain from both ingestion and metabolism can exceed the body's maximum excretory capacity. This is especially true if solute excretion is also reduced.
How does solute excretion limit water excretion?
Think about it. To excrete water, you need to excrete some solute to carry it out, even if the urine is very dilute, like 50 mg. If your total daily solute excretion is low, say, in elderly individuals with poor food intake, maybe only 400 millimoles per day, then even at the maximum dilution of 50 mg, the maximum urine output is only 8 liters. 450 equals 8. Compare that to the 18 liters possible with higher solute excretion. So low solute intake profoundly reduces the capacity to excrete water, making those individuals much more vulnerable to hyponatremia if they drink too much free water.
That's a really important clinical point. Okay, we've gone deep on water. Let's shift focus to sodium. Sodium chloride is the major determinant of ECF volume, right? So what does this all mean for controlling our overall fluid volume?
A critical insight, as we touched on earlier, is that changes in total sodium balance primarily alter the ECFV, the extracellular fluid volume, not its osmolality.
Because the AVP thirst system jumps in to fix the concentration.
Exactly. That system is incredibly efficient at keeping osmolality stable. So if you acutely add NaCl without water, osmolality tries to go up, AVP thirst kick in, water is ingested or conserved, osmolality comes back to normal. But now you have more water diluting that extra salt, so your ECF volume is increased.
And the reverse if you lose salt.
Yeah, precisely. And practically, we can monitor these dynamic ECF volume changes by tracking body weight. 1 liter of ECF is very close to 1 kilogram of body weight.
The kidneys are the main route for getting rid of salt.
Overwhelmingly, only about 10% is lost non-renally, sweat, feces. So the kidneys are profoundly important in regulating ECFV. Normally, they maintain euvolemia constant ECFE by meticulously matching NaCl excretion to dietary intake.
What happens if intake and excretion don't match?
If sodium intake chronically exceeds excretion, ECFV increases volume expansion. If excretion consistently exceeds ingestion, ECFV contracts. But the kidneys have an astonishing adaptive capacity. They can vary sodium excretion from maybe 10 ml a day on a super low salt diet to over 1,000 ml a day on a high salt diet, all while keeping ECFV relatively stable.
You mentioned earlier this isn't just about total volume, but effective volume. Can you explain effective circulating volume, ECV, again?
Right. ECV isn't a distinct measurable compartment like ECFE. It represents the portion of the ECF, mainly within the arterial system, that is effectively perfusing the tissues. Think of it as reflecting the activity of the body's volume sensors in the vascular system.
Why is this concept so important?
Because it explains paradoxical situations. The classic example is congestive heart failure. These patients often have a massively increased total ECFE. They're edematous, fluid overloaded.
Right.
But their ECV is paradoxically decreased due to poor cardiac output, low arterial filling pressure, maybe capillary leak. So the body's volume sensors perceive this low ECV as if the ECFE itself is contracted.
And the kidneys respond
by inappropriately retaining more sodium chloride in water, making the fluid overload even worse despite the clinical picture. Understanding ECV is key to understanding that pathophysiology.
Okay, so if the kidneys are the effectors adjusting salt and water, what are the upstream sensors detecting these changes in ECV? And what signals do they send?
These sensors are collectively the vascular volume receptors, or baroreceptors. They fundamentally respond to pressure-induced stretch in the walls where they reside, blood vessels, heart chambers.
And they're in 2 main circuits.
Yes. First, the low-pressure cardiopulmonary circuit. Receptors in the atria, right? Ventricle, large pulmonary vessels. These respond mostly to the fullness or distension of the vascular system. They send signals to the brainstem via the glossopharyngeal and vagus nerves.
What do those signals control?
Their activity inversely modulates sympathetic nerve outflow and AVP secretion. Less filling means more sympathetic activity, more AVP. More distension means less sympathetic activity. Like the osmotic system, it takes about a 5% change in volume pressure to trigger a significant response.
And the heart itself releases hormones.
Crucially, yes. The cardiac atria release atrial natriuretic peptide, ANT, when they distend. ANP acts to lower blood pressure and powerfully increases renal NaCl and water excretion. The ventricles produce a similar hormone, brain natriuretic peptide, BNP.
Okay, that's the low pressure side. What about the high pressure arterial circuit?
Baroreceptors there are in the aortic arch, the carotid sinus, and importantly within the renal afferent arterioles themselves. The aortic and carotid ones send signals to the brainstem, influencing sympathetic outflow and AVP, similar sensitivity to the low-pressure ones.
And the kidney senses its own pressure.
Yes. The juxtaglomerular apparatus, JGA, specifically the afferent arteriole, acts as a local pressure sensor. Reduced perfusion pressure directly triggers renin release from the JG cells. Increased pressure suppresses renin release.
And that's clinically relevant in things like renal artery stenosis.
Exactly. If a renal artery narrows, perfusion pressure to that kidney drops, stimulating massive renin release from that kidney. This boosts systemic angiotensin II, causing hypertension. Interestingly, the high angiotensin II then inhibits renin release from the other, healthy kidney via negative feedback. Managing it often involves blocking RAAS or fixing the stenosis.
So when these sensors detect an ECV change, they trigger a complex array of signals aimed at the kidneys.
Right. During ECF expansion, the signals work together to increase NaCl and water excretion. During ECF contraction, they recalibrate to powerfully decrease excretion, conserving fluid.
What are the key signals involved?
The main players are the renal sympathetic nerves, the pivotal renin-angiotensin-aldosterone system, RAAS, the natriuretic peptides, ANP, BNP, and arginine vasopressin, AVP.
Let's break down the renal sympathetic nerves first. What happens when ECF contracts and sympathetic activity increases?
Increased sympathetic tone hitting the kidneys has 3 crucial effects. First, it constricts both afferent and efferent arterioles, but more so the afferent. This lowers glomerular capillary pressure, decreasing GFR and the filtered sodium load.
Okay.
Second, it potently stimulates renin secretion by the JG cells. And third, it directly simulates NaCl reabsorption along the nephron, especially in the proximal tubule where most sodium is reabsorbed anyway.
So the net effect is?
A substantial decrease in NaCl excretion. It's a rapid response to conserve volume. During ECF expansion, the reverse happens. Less sympathetic activity promotes salt and water loss.
Now the big one. RAAS, renin-angiotensin-aldosterone system. How does that start?
It starts with renin, an enzyme released from those juxtaglomerular JG cells in the afferent arterioles. Renin release is stimulated by three main things: the renal baroreceptor sensing low pressure, beta-adrenergic receptors on JG cells being hit by sympathetic nerves, and the macula densa sensing low NaCl delivery in the distal tubule.
And renin secretion has that weird calcium regulation.
Yes, very unique. Renin secretion is paradoxically stimulated by a decrease in intracellular calcium in the JG cells, mainly controlled via the CAMP pathway, opposite to most hormone release.
Okay, renin is released. What's next in the cascade?
Renin cleaves angiotensinogen, a protein made by the liver, into angiotensin II. Angiotensin I itself isn't very active. It's then rapidly converted to the highly active angiotensin II by ACE, angiotensin-converting enzyme.
Where is ACE found?
It's widely distributed on vascular endothelial cells, but the lungs and kidneys are major sites. ACE also breaks down bradykinin, a vasodilator, which is relevant for some drug side effects.
And angiotensin II is the real workhorse here. What does it do?
It's incredibly multifaceted. 1, stimulates aldosterone secretion from the adrenal cortex. 2, powerful vasoconstrictor, raising blood pressure. 3, stimulates AVP secretion and thirst. 4, directly increases NaCl reabsorption in the proximal tubule, thick ascending limb, distal tubule, and collecting duct. Really pushes salt retention.
And aldosterone, the final hormone in the chain.
Right. Angiotensin II triggers aldosterone release from the adrenal cortex. Aldosterone is the main mineralocorticoid. It fundamentally stimulates NaCl reabsorption, mainly by increasing the abundance and activity of enase, the sodium channel on the apical side, and then Na plus AKP plus ATPase, the pump on the basolateral side, in the principal cells of the distal tubule and collecting duct, and also some effect in the thick ascending limb.
So the overall effect of RAAS activation, like during ECF contraction, is decreased salt excretion.
Powerful decrease, yes, leading to salt and water retention. Conversely, RAAS suppression during ECF expansion leads to increased salt excretion.
What happens clinically if aldosterone levels are wrong?
Big problems with sodium balance. Hypoaldosteronism means less NaCl reabsorption, more salt loss, ECFE contraction, and importantly, high potassium, hyperkalemia. Hyperaldosteronism means increased NaCl reabsorption, low excretion, ECFV expansion, and low potassium, hypokalemia. You also see compensatory changes in the other systems like suppressed sympathetic tone, renin, AVP, but high natriuretic peptides in hyperaldosteronism.
Okay, so RAAS retains salt and water. What's the counterbalancing force?
The natriuretic peptides, ANP from the atria, BNP from the ventricles, and urodilatin from the kidneys themselves. They're released during volume expansion or heart failure and act synergistically to powerfully antagonize RAAS effects.
How do they promote salt and water loss?
Through several mechanisms. 1, increased GFR by dilating afferent arterioles and constricting efferent ones more sodium filtered. 2, inhibit renin secretion, suppressing RAAS source. 3, inhibit aldosterone secretion directly and indirectly. 4, inhibit NaCl reabsorption directly from the collecting duct, partly via cGMP pathways. 5, inhibit AVP secretion and its action on the collecting duct, reducing water reabsorption.
So they're basically the get rid of salt and water signal.
Exactly, a crucial physiological counterbalance. And AVP, of course, when elevated during ECF contraction, decreases water excretion to help restore volume.
Okay, let's try to put it all together now. How does the kidney as a whole integrate all these signals to maintain EC/NV? It seems incredibly complex.
It is complex, but beautifully orchestrated. Remember the scale. About 67% of filtered sodium is reabsorbed proximally, 25% in the thick ascending limb, and the rest fine-tuned in the distal tubule and collecting duct.
So even tiny changes matter.
Hugely. A mere 1% decrease in total sodium reabsorption could mean losing 2.5 liters of fluid a day, a massive hit to ECFE. Underscores the need for precision.
Under normal conditions, euvolemia, where is the fine-tuning happening?
Primarily in the distal tubule and collecting duct. They adjust reabsorption to match dietary intake. but their capacity is limited, so the proximal tubule and loop must efficiently reabsorb the bulk, about 92% first, delivering a relatively constant 8% of the filtered load distally.
How is that constant delivery maintained?
Through several coordinated mechanisms. GFR autoregulation keeps filtration stable despite pressure changes. Glomerulotubular balance ensures proximal reabsorption adjusts proportionally if GFR does change. And the loop's reabsorption increases if sodium delivery to it increases, load dependency. The aldosterone-sensitive distal nephron then makes the final adjustments.
Okay, now let's trace the integrated response to ECFE expansion. What signals are firing?
When ECF volume expands, the volume sensors trigger signals to increase excretion. So decrease sympathetic nerve activity, increase natriuretic peptides, ANP, BMP, urodilatin, inhibition of AVP secretion action, and suppression of the entire RAAS, less renin, less AIM2, less aldosterone.
How does the nephron respond to that signal cocktail?
3 main things happen. 1, GFR increases. This is due to less sympathetic constriction and the direct effects of natriuretic peptides on glomerular arterioles. More GFR means more sodium filtered. 2, sodium reabsorption decreases in the proximal tubule and loop of Henle. This is due to less sympathetic drive, less Ang2, and also changes in paratubular Starling forces that favor less reabsorption. Importantly, glomerulotubular balance is overridden here, allowing net excretion. More NaCl gets delivered distally.
And third,
sodium reabsorption further decreases in the distal tubule and collecting duct. This is driven by the increased load arriving there, plus less Ang2, less aldosterone, and the inhibitory effects of natriuretic peptides.
result.
Marked increase in both NaCl and water excretion, restoring euvolemia. This response takes hours to days, depending on the expansion size.
Finally, the integrated response to ECFE contraction. What signals fire then?
Here, the sensors detect low ECV and send signals to reduce excretion. So increased sympathetic activity, powerful RAAS activation, more renin, Ang2 aldosterone, inhibition of natriuretic peptides, and strong stimulation of AVP.
And the nephron's response.
Again, integrated actions for conservation. 1, GFR decreases, mainly due to sympathetic constriction of arterioles, reducing the filtered sodium amount.
Conserving fluid right from the start.
Exactly. 2, sodium reabsorption increases in the proximal tubule and loop of Henle, driven by more sympathetic activity, more Ang2, and favorable Starling forces promoting reabsorption.
Okay.
3, sodium reabsorption is further enhanced in the distal tubule and collecting duct, partly because less sodium arrives, but also due to the powerful effect of elevated aldosterone. 4, water reabsorption is dramatically enhanced by the high AVP levels, stimulated by volume sensors and Ang2.
So the net result is?
Significant reduction in both NaCl and water excretion. The body retains salt and water proportionally, restoring euvolemia, and again, crucially, glomerulotubular balance is overridden to allow this net retention.
Wow, what an incredible journey into the sophisticated internal world of your body. From, the precise molecular machinery of AVP opening and closing water channels to the grand interconnected symphony of RAS and natriuretic peptides, it is unequivocally clear that your body is constantly performing this exquisite, complex balancing act.
It really is. Keeping that internal environment perfectly tuned, it's a profound testament to the sophistication and adaptive capacity of human physiology.
Yeah.
And what truly stands out for me as we aim for this deep mastery isn't just the brilliance of each individual part, but the sheer redundancy and interconnectedness. Multiple systems, multiple checks and balances.
Fail-safes.
Exactly, fail-safes. Ensuring that even under extreme stress, the body fights tirelessly to maintain homeostasis. This comprehensive understanding really highlights why digging into these mechanisms with such depth is absolutely critical, not just for clinicians, but for anyone wanting to truly master the intricate workings of their own biology.
So as you reflect on this deep dive, what connection, what intricate detail, or maybe what integrated pathway stands out to you most as you strive for that professional mastery? Next time you just reach for a glass of water, or maybe feel that innate craving for something salty, you'll possess a whole new, profound appreciation for the invisible, hidden world inside you, meticulously controlling every single drop of fluid and every single grain of salt.